Going to be on exam 3, and therefore the last thing that'll be on the final, this idea of complementation. It's an important genetic test. And we don't get to talk about genetic tests much because we don't actually do genetics. We don't have a genetics lab, although you do genetics and other things. But still, complementation. Not to be confused with DNA complementation or complementarity, but it's similar. Here, the idea of complementation is, can you give an organism something that it is missing? And typically, it means there's a mutation affecting a gene, making that organism unable to do something. And so it needs a replacement part from another organism. And there are a couple different ways that we can do complementation in the lab. The classic example we'll talk about in a second is using diploid organisms to make gametes and then combine those gametes together, see if they complement each other, or at least we get complementation. With bacteria, because they're haploid, we do some other things. Typically it means either using plasmids or other methods to introduce DNA or add DNA in different ways. You can use the Cas9 system to add something new as a way to see if complementation is possible. So there's also different ways we can add things to microorganisms or to other organisms to see, can we fill in the gaps? That's what, again, complementation is about, filling in the gaps. Now, most oftentimes we represent complementation in a vein of a chemical pathway. Like shown here, a multi-step pathway, where A is used to make E, and E is some essential nutrient or something. So typically we're testing to see if mutant 1 and mutant 2 are the same or are they different. And so the original sort of idea of complementation is to compare one mutant to another. And in these two situations, you clearly see the number of mutant 1 is different than mutant 2. Our general path is here. But if you don't know what's going on, you have no idea. So what you do is you mix 1 and 2 together and see what happens. And we have some rules to sort of think about. But if complementation happens, then number 1 benefits and number 2 benefits, then basically you get this substitution effect happening, where number 2 is giving number 1 the missing function, and number 1 is giving number 2 the missing function. So together they are a whole. We say, a real boy. Don't give us things. They used to talk about Mr. Potato Head here, but people just didn't really understand Mr. Potato Head. Raise your hand if you know Mr. Potato Head. Did you have a real one or a fake one? Let me ask, what's a fake one? It's a real potato with just made up parts. Two scientists, the last two scientists, the very last two scientists that you're going to hear about in this class, Beadle and Tatum. They proposed a very important idea in molecular biology that is now not exactly true, but still the one gene, one protein idea. Again, what we base most of our molecular biology on is one little piece of DNA makes an RNA, which makes a protein, and that should be a sort of a one-to-one thing. But these days, we know that there's alternative splicing and all sorts of weird things that can happen sometimes. So it's not always true, but generally speaking, one gene makes one protein. And Beadle and Tatum go hand in hand with this idea of complementation, trying to identify what's wrong with a mutant without having DNA sequencing or the cool tools that we have today. Just for perspective, this was in 1941. So they remember, is this before or after Watson and Crick proposed the three-dimensional structure of DNA? And before, so it was 1950s that happened. Pretty early on in sort of the scientific world, this idea came about. Might need to know that Beadle and Tatum studied a fungus called Neurospora, which makes it a diploid organism, so it's very useful in this process because you can take a diploid fungus and make spores, which are haploid, and you can combine spores from two different mutants together to remake a diploid organism and see what happens. And so they used UV light to cause mutations or X-rays. We covered UV light last time because they may know what X-rays will do. for us, what is the one important thing that X-rays would cause? And DNA breaks. Double-stranded DNA breaks in the genome. But it turns out that X-rays also cause just regular mutations, but for us, ionizing radiation like X-rays and gamma radiation should be the main cause of double-stranded DNA breaks. And like most scientists, they were studying one particular thing, and they were looking at the ability to make the amino acid arginine. So they make mutants that can't grow without arginine, so they have to add arginine to the medium. So if complementation happens, you don't need arginine anymore because you're getting something you're missing. Now, when we think about complementation, we're looking for an end result. This is where students get somewhat confused. Complementation means that growth has occurred. Because complementation should give you something you're missing. Like A, adenine needs T, because they go together. G needs C because they go together. So complementation, a positive result for complementation means growth. Nothing else is needed. We have everything we need to survive and exist on our own. And hopefully the reverse is pretty easy to identify. No complementation means no growth. Or at least the result of no complementation is no growth occurring. This part is not the hard part. The hard part the students struggle with is, what does this mean? That's where we get into this slide here. So on this first slide here, you can see we're combining together two different mutants, mutant 1 and mutant 2. And for illustration, you can see that they're different. We've got a mutation in this orange gene in one, and a mutation in the purple gene in the other one. If you combine these together, you get the diploid organism back. And so you have one functional orange and one functional purple, so we're good. We can grow. Complementation occurs and growth happens. The only reason that this happened is because both mutants had different genes affected. If they had the same gene affected, you're not getting something you're missing. So complementation happens when you get the thing that you are missing. So if our mutants are different, then complementation is a possibility. And different genes have to be affected. If you look at the board on this slide, mutant 2 and mutant 3, you may notice that little X is in different locations. As we're going to see if you go forward, that doesn't matter. It's not where the mutation is relative to the other, it's which gene is mutated relative to the other. So if we have the same gene mutated in two different mutants, no complementation can occur because we're lacking the exact same function in both those organisms. So again, in this case here, one gene is mutated in mutant 1, a second gene is mutated in mutant 2. So combine them together, you get one good copy of one good copy of the other. Again, if you have two different mutants or two mutants with the same gene affected, then you still don't have a functional purple. Whatever purple is, you don't have it. So that is a case where no growth occurs, meaning complementation didn't happen. Again, the interpretation of no complementation means the same gene is affected in two different mutants, but if we do get growth, meaning complementation happened, different genes are affected. And what we're trying to do with this complementation idea is identify which mutants have the same gene affected and put them into a group together, and anything that's not the same is in a different group by itself. So mutants that the same gene is affected are going to go into group together. This is the other thing that drives students a little crazy, because negatives go together. Today you think positives should go together, but it doesn't, unfortunately. We'll go through some examples and see how everybody does with it. But if you're different, then you're different groups. So we're trying to build what we call complementation groups. Things that have the same gene affected will be in a group together. Things that have different genes affected will be in different groups. And we see this data represented in a table that looks like this. And we're trying to analyze these positives and minuses and what these mean. So we take mutants. In this case, the easy case is we take a mutant 1 and we make gametes. And take mutant 2 and make gametes and fuse them together with fertilization and see what happens. Do they grow or do they not grow? That's what positive means. If they grow, they have positive. If the negative means they don't grow. And what you want to notice is your diagonal is the same mutant bred to itself. And so is mutant 1 lacking the same thing that mutant 1 is lacking? Does 1 lack the same thing as 1?
Yes.
Is 1 the same as 1? Yes, right? 1 is the same as 1. And so both are missing the exact same thing, so they shouldn't complement each other. Which means the same gene is affected. So again, this is our sort of clue. When you mix something with itself, you should not get any growth, because you're not getting the thing you're missing. And if you mix it with something different, like here you mix number 1 and number 2, you get growth. That means number 1 and number 2 are different, because now you're able to grow, whereas before you weren't. But the positives mean number 1 and number 2 have different genes affected. So 1 and 2 can't be in the same group together, because they're not the same thing. But here, number 1 and number 5 will go together, because they don't complement each other. So trying to build these groups. We're always looking for the negatives. The negatives tell us what's going on. The positives we can basically ignore. And so in this example here, it's a pretty easy one. There's only 2 groups. Number 1 and number 5 go together because 1 and 5 do not complement each other. But 2 complements 1, 3 complements 1, 4 complements 1, 6 complements 1. And the same thing is true for 5, 2, 3, 4, and 6 will all complement number 5. And 2, 3, 4, and 6 are in a group together because none of these complement the others. So they all go into one group together. Again, we group them together because they have the same mutated gene. That's what we're trying to do with complementation tables. Which mutants have the same gene affected? This is a simple example. Typically they're more complex, like this one as an example. So here we go. You can look at this 2 ways. Don't get confused by which diagonal is shown. Sometimes it looks like this. Sometimes it looks like this. Sometimes it looks like this. Sometimes it looks like that. Sometimes it looks like this. Sometimes it looks like that. It doesn't matter which side, top or bottom, you still get the same thing. To read this, you need to go both directions. You can't only go one direction. That means you either go sideways and then downwards. Or downwards and then sideways, depending on which orientation you have. One really important rule is everything needs to be in a group. How many mutants does it take to make a group? How many does it take to make a group? 1. Because each group represents a different gene being affected. And so a group of 1 is still a group that's different than all the other groups. That's the first important thing that students forget. A group of 1 is still a group. Things cannot be in different groups. That's the other important rule. I see a lot of times when people will say that A and F will be in a group together. Because they're negative, it means they don't complement. So A and F have the same gene mutated. And then later on they'll say F is in a group by itself. this way and say F. Yeah, okay, I'm done. F. I don't know what they're doing. Students are weird sometimes. If you say that A and F are in a group together, you can't say that A is separate later on, or F is separate later on. Or another case we'll look at here in just a second. I've done one up for you, A. We go across, then we go down. So you hit the diagonal, and then you go the other direction. What about for B? Is B, what do we know about B? What do you tell me about B? What do we see with B?
B is on its own.
And why is it on its own?
Because it only negatively affects itself.
Yeah, there are no other negatives other than when B is with itself. So A and B complement each other, meaning they can't have the same gene affected. So A and B are not in the same group together. B and C complement, so same gene, or different gene, can't be the same group. And all these are positives. And positives mean different genes affected, so they can't be in the same group together. So now we have A and F in one group, and B in a second group. What about for C? What do we know about B and C? Is it by itself, or is it with other things? B and G. So again, if you go across, A complements, B complements, so those are different. You go down, so you get no complementation. So C and D have the same gene mutated, which is why they don't grow when they're mixed together. And then G is also in the same category. So C, D, and G are in the same group together. Okay, this is where typically students will make a mistake. They'll say C, D, and G will be in a group together. And then later on, we'll say that D and G are in a group together. And sometimes say that G is in a group by itself. So they're doing some weird type of interpretation I really don't understand. Everything has to be in a group, and everything you have to be in. If you're group, you're group. So put these in groups. So we got A and F. We got B. We got C, D, and G. There's only two letters left. I choose on one letter left, right? E. What do we see with E? Does E do anything? And E has all positives also. So E is in an own group by itself. So one group that's A and F, one group that's B, one group that's C, D, and G, and one group that's E. That's 4 groups. I won't talk on this next one. See if you can figure it out. And this is your roadmap, so this is what we want to remember. We're looking for no growth, because that tells us if things are in the group together. We'll take just a second or two or a minute or two. There'll be a few more of these posted on the lecture reviews. If you want, I can post a quiz for you to take. That's 4 different points. You think that would be helpful? Does anyone want to take a quiz rather than just doing the problem on the lecture review? As an answer. That's ambiguous, unfortunately. Saying sure to the first part, or sure to the second part, or sure to both? See, I don't know. So, what do we think? What do we got? Who tell me in one group? L?
A, C, and G.
So A, C, and G. Yeah, so if you go across, you go down to the diagonal and then go across, you see that A and C don't complement, therefore the same gene was affected, so they're in the same group. And then go across and pick up G again, so yes, A, C, and G are in one group together. Who can tell me in another group? B and F. Again, so we'll start with B and then go down to the diagonal, and we see that there's a negative here with F. Again, the negative means no growth, no complementation, same gene affected. Therefore, B and F are in the same group together. Who can tell me in another group?
D and E. D and E.
D and E. All right, we've already done C, so don't worry about it. But you should see that things match. So C, A, and then G. That's always good. Make sure everything's correct, because sometimes I screw up. You don't need to tell me when I screw up, because that throws everything off. But D and E, yes, and you see only one negative there is D and E. Have they accounted for all the letters? So A, C, and G, and B, and F, and D and E. So only 3 groups here. You want to do another one? Or do you want to leave?
Do one more.
Well, those of you who want to leave, you know where the door is. Those of you who want to stay, you know where your seat is. So let's pick one from last semester. What exam was it on? Let's see. I think it was on exam 3 again. Let's see. No, it wasn't. Wait. No, it wasn't. Let's see. Wait, no. Let's see. Exam 1. No. I think it's exam 2. It's good. Yeah, I think it's gotta be in there. It's gotta be in there. Let's see. Don't look. Don't look. There it is. Okay, here we go. Is it big enough? Should I make it bigger?
A little bit, please.
Stop fiddling with it, Dr. Bridget. Okay. Does anybody know why we don't need the top half?
Because you already did it the other way.
What's that?
Like you already did it the other way.
It's just a mirror image. Yeah, so we don't need to see both halves because they should be exactly the same regardless of how you do it. But the idea is that if you mix A with B and then mix B with A, you should get the same result. But sometimes order of addition does matter sometimes, but not for this particular case. Sometimes we use numbers instead of letters, not to throw you off, just to be a variety. Say. So instead of A through H, it could be 1 through 9, or 1 through 8, or 1 through 10. Don't let that throw you off. Anybody still working? Unless somebody has answered. Do you want to go ahead?
I have a question.
Okay, question.
Wait, every letter doesn't have to be used, does it?
Everything has to be in a group.
Oh.
That group could be 1,
Oh.
but if you don't
Okay.
answer for every single letter, then you're telling me that you either don't know what to do with that thing, or you forgot to deal with it,
Okay.
or you just ignored it, or you don't care enough about it.
Okay.
Either way, I guess, just irregardless of all of those things. I love just irregardless. It's nonsense. It's the point of deduction if you don't account for all the things that are there. So yes, well, that's why I say first, everything has to be in a group.
Okay.
That group could be 1. But everything has to be identified in some way or another. If you just don't do it, then it means you're not answering fully. So you get credit for what you did and lose points for what you didn't do. So if you want to make hands up before, but didn't answer. A and H. Yeah. Pretty easy. Anybody else want to give a go that hasn't done yet?
B and F.
B and F? Yeah, B and F. One.
C and G.
C and G. Yeah, C and G. Is there any letters that haven't been accounted for yet?
D.
Okay, are D and E together or are they separate?
Separated.
Okay. Yeah, it's not that hard, is it? I mean, if you understand the concept, it's very easy. But it's easy to get sort of lost and do these go together or do they not go together? Is a positive good or a positive bad? And here, positive is bad. Negative is good. We're looking for the negatives rather than the positives. And I always say, look to the diagonal if you forget. What am I trying to see? Because if you're at it to yourself, you should never grow because you have obviously the same mistake as yourself. Mr. Potato Head missing an ear needs to find another potato head box with an ear, not another box with missing an ear. So they go garage sale hunting for an extra ear or extra arm they lost. Let's leave the rest of it. Now you can buy the whole thing, right? Replace that one missing thing, you gotta buy the whole thing. Sucks, but that's the idea of complementation. Okay then. Does anybody want to ask any questions about exam 3 today? Or quiz, whatever it is? 8, 9. And then
On the quiz due Monday, it goes from mitosis to, what was the ending word of it?
mitosis.
Just mitosis.
Well, mitosis, because everything we talked about in cancer cell cycle regulation is mitosis as well. We talked about the G1S phase transition, which is mitosis, and then the G1, G2, so everything is wrapped around mitosis. So yes, the steps of mitosis and the cell cycle, and all the regulatory components we talked about are the quiz. Today's stuff is not on the quiz. Repair mechanisms are not on the quiz. Regulatory methods like induction, repression, RNAs, those aren't on the quiz because that was the last quiz. What are the stages of mitosis? What are the stages of the cell cycle? And what proteins control the movement from one stage to the other?